full screen. Okay. So before we get into action potential, I want to briefly review what we did before. So at rest is polarization, getting more positive, depolarization. In order to start an action potential, the neuron has to depolarize to a certain point called the threshold. This top picture shows a neuron depolarizes a little bit, but that dotted line is the threshold. It doesn't get there, so it's just going to gradually repolarize and go back to resting. In this one, it's getting a stronger stimulus. That depolarizes the trigger zone enough to reach that threshold. Then this big spike is the action potential. So here's what you'll see in an action potential. In one little region of the axon, you'll see this specific pattern of changes to membrane potential. So first, it depolarizes. That's shown in yellow. Then it repolarizes. That's shown in purple. I'm going to put a dotted line here to show where resting potential is. Before it, so instead of just going back to resting, it's going to hyperpolarize. And then it returns to resting. Okay, and this dip was probably further than it really would be, but just to show you the idea. That's your pattern. An action potential is this area of membrane right here depolarizes, repolarizes, and hyperpolarizes before going back to rest. What makes it change are movement of ions through voltage-gated channels. They're opened when we hit the threshold. Between these red marks, sodium channels are open. Sodium's more concentrated outside the cell, so where's it going to go? It's going to go inside. Sodium is positive, that makes the inside more positive. Between these marks, well, really this one should be here, the potassium channels are open. Which way does potassium want to go? It wants to go out. So we let one positive thing in, and that depolarized. At this peak where the inside is as positive as it's going to get, sodium channels close, the potassium channels open. And I'll mark here threshold, sodium channels open. Now, the potassium channels start closing when we get back to threshold, but you know how some doors are whack and some of them are. The potassium channels are the slow ones. They actually start opening at the same time as the sodium ones, but it takes them until here to be open enough for potassium to get through. They start closing here, but it takes them until here to actually get shut. So more potassium is sneaking through. And that's why instead of just letting out the same amount of potassium we let in and balancing the charge, we're letting out extra and hyperpolarizing. So here's neuron at rest. Here's where it's depolarizing. The purple is the sodium coming in from outside. And then here's where it's repolarizing and hyperpolarizing. The potassium is the pink ones going out. Okay, on how the action potential works. Now, how this turns into a message that goes the whole length of the axon and can stimulate the next cell is called conduction. You've got a stretch of axon, you get an action potential here, that's going to cause the next section to have an action potential. And that will go all the way down the line. The way it works is your voltage-gated sodium channels are along the whole length of the axon. And once you've opened the first set, the sodium that comes in through them and depolarizes that section spreads out a little bit and takes the next set of channels to threshold. It's kind of like knocking over dominoes, where you only have to hit the first one, and if they're lined up correctly, which the channels are, they're each going to knock over the next one down the line. You could also think of it as you've got a hallway full of doors. Sodium comes in the first door, takes a step, and opens the next door. And then another sodium comes in that one, goes over, opens the third door. And that will go all the way down to the axon terminals. So here's that same video we had last week. I'm going to get it ahead to get some more sound. The first part was the resting potential. We watched that last week, and now we'll look at how the action potential works.
The resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
So it's not a 6-minute video, just for some reason it has the same thing twice. So as he said at the end there, for our action potential, we've let sodium leave, or come in. We've let potassium leave, and we need to use our sodium-potassium pump, our active transport, to put them back on the side they need to be on. So So that's your action potential. Your conduction makes it happen one little stretch at a time, all the way down to the end. Now, if we have myelin, it makes the message go faster. Here's how that works. It's kind of like insulation. Ions can't cross where the myelin is. So when the sodium comes in, instead of just going a short way and opening another channel, it flows faster and further, opens a channel in this spot between myelin, which is called a node of Ranvier. It might be Ranvier, but I learned French, so I say Ranvier. And then the sodium from that one will come in, go to the next node. So if you're just measuring the voltage throughout the axon, you're going to see action potential here and then here. And so it looks like it's jumping. That's why we call it saltatory. Anybody speak Spanish? What's the word for jump? Salta, yeah. Spanish comes from Latin. Most anatomical and physiological terms come from Latin. So this is called jumping conduction, because instead of action potential here, 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 it's here, here, here. It gets to the end faster because the ions can just come in and run, not that they have legs, but you know what I mean, before they have to stop and open the next door. And this is the first class where anybody has actually known the answer to that. Spanish. So you would see something like this where your electrical potential changes in this node, and then this node, and then this node, and the arrows indicate the sodium traveling. So it's still continuous with ion flow, but the changes in electrical potential are spread out. That would be like if you had your domino go down a slot to hit the next one. So, any questions on action potential? Localized or conduction? Okay. Now, when our action potential gets to the synaptic knob, it can't just keep going to the next cell because they're not touching. So instead, when it gets down to the end, instead of letting more sodium in, it's going to stimulate vesicles of neurotransmitter to go to the surface. Now, you'll see in the textbook and some videos that the way that works is it lets calcium in instead of sodium. And instead of just changing the charge, the calcium activates the vesicles. I don't need you to remember the calcium, just that the action potential stimulates exocytosis, the neurotransmitter is released, and then the postsynaptic cell has receptors for it. In the case of the neuromuscular junction, those receptors were actually chemically gated channels. When the acetylcholine binds, they open and let sodium into the muscle cell, which causes it to have an action potential. A lot of neurotransmitter receptors are voltage-gated or chemically gated channels. If they were to say let potassium out, that would hyperpolarize the cell, and that's one way you could have an inhibitory stimulus. The neuromuscular ones let sodium out, that depolarizes, brings them toward threshold, so that's excitatory.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine.